presentation.
I'm sure my career is much less interesting than Julio has highlighted, so thank you very much for the invitation to come and speak. And I'm going to be talking about the discovery of potent selective small molecule inhibitors of a BET family of bromodomains and how we believe that this demonstrates the tractability of this epigenetic reader class for drug discovery. So I'm going to start the talk with a very brief introduction into epigenetic readers and how they participate in multivalent protein-protein interactions. But our story really starts with the discovery of small molecule ApoA1 activators, and how we were able to unravel their mode of action was through, unexpectedly, bromodomain inhibition using a number of biophysical and structural techniques, and how we believe that this has application broader than to ApoA1 and highlights opportunities for structure-based drug design and a system-based approach for a class of targets people have thought were untractable, which is these bromodomains. And looking beyond ApoA1, what I'm going to show you is the applicability of these compounds outside of their initial area of therapeutic discovery. And we're investigating a number of these, including cancer and viral responses. But I'm going to just highlight their possible application in an immunoinflammatory context, where these compounds have rather a profound and dramatic effect in a very acute inflammatory model, an LPS-induced shock model in mice. So one mechanism of epigenetic gene regulation is via the post-translational modification of the N-terminal and exposed sites of histones. There are many types of modifications that have been reported on a number of amino acids. Most of these are dynamic and reversible, and they can be small modifications such as acetylation, methylation and phosphorylation, or larger modifications such as stimulation and ubiquitination. One simplistic way that these post-translational modifications can have an effect is by directly impacting on the physical properties of the individual nucleosomes. For example, acetylation lysines neutralize charge, and that can affect the structure of the chromatin, making it more accessible for transcription factors. A more powerful way that these post-translational modifications can elicit effect is by first the recruitment to the site of the modification specialized read domains that sense that the modification has occurred, and this can lead to propagation of downstream recruitment events and also enzyme activation. Now, post-translational modifications rarely occur in isolation. What you tend to get is complex and combinatorial patterns of modification that's sometimes known as the histone code. Similarly, read domains rarely occur in isolation. They can appear concatenated as part of the same protein, or they may be brought together by virtue of protein-protein interactions, and it's then the complexes that recognize this pattern of modifications. As I mentioned, histone acetylation is normally associated with gene activation. So there are enzymes, Histone acetyltransferases, that put on this acetylation, and other enzymes, histone deacetylases, that take off these modifications. HATs and HDACs may be familiar to you because they've been widely pursued in the pharmaceutical companies as targets for possible disease intervention. Maybe less familiar to you are the proteins, the small proteins that sense the presence of the acetylated lysine. And these are called bromodomains, and they can appear singly or joined together sometimes in some proteins. And there are actually 58 of these in the human genome. And just like HATs and HDACs represent convenient control points for disease intervention, so do bromodomains. But till recently, they've been widely ignored by the pharmaceutical companies because they were considered to be poorly tractable for small molecule drug discovery. So if we examine some of the criteria that people use normally to judge whether targets are tractable for pharmaceutical pursuit, you can really understand why this might be. Reader domains often bind their post-translationally modified peptides rather weakly. Bromodomains are notorious for this, so you can often get a recognition peptide that only shows 100 micromolar affinity, sometimes millimolar affinity. So if you're looking at protein-protein interaction, people tend to look for hotspots, places where if you put a small molecule, you can really disrupt that interaction. I mean, for bromodomains, it's more like an iceberg rather than a hotspot. What you also have is multivalency in these protein interactions, so multiple points of interaction. So if you imagine trying to target that with a small molecule, you might be able to disrupt one site, but only to have it kind of spring back, where the other site is still kind of tethered, the so-called tethered ligand effect. These proteins are normally within the context of megadalton protein-protein DNA complexes. So not only do you have this multivalency, but a question that you might ask is, well, is there any redundancy in the system? So would inhibiting just a single interaction be enough for biological efficacy? People may know that protein-protein interactions are generally considered to be poorly tractable. Given now what we know about these epigenetic reader proteins, it's really not surprising people have shied away from these as pharmaceutical targets, focusing instead on what seems to be, on the face of it, more tractable epigenetic enzymes that have sort of well-defined pockets. Fortunately for us, we weren't burdened with that knowledge, and in total ignorance, what we were looking for in 2001 were ApoA1 upregulators, small molecules, as a target for dyslipidemia. We didn't really have a target in mind because we didn't know how to do this, and so what we set up was an ApoA1 upregulator assay using a luciferase readout in HepG2 cells. And we were successful in identifying several lead series, including a benzodiazepam series as exemplified by this core structure on the right here. Our medicinal chemists in Lazy Lee were very talented and successful and optimised these molecules to candidate selection without knowledge of the molecular target. And in fact, because there were diverse chemotypes, potentially molecular targets of these interactions. Naturally, what we'd done was taken these compounds and extensively profiled them against all our internal and numerous external assays, but to no avail really, and we had no clue as what the target or targets of these molecules were. But what we did have was a lot of chemical equity, so we embarked upon a chemoproteomics study. So taking compounds like these BZD compounds, where one enantiomer is active and another one isn't, we're able to couple these onto matrix, challenge them with lysates from relevant cell types, and what we were hoping to see was retention of some proteins on the active matrix that were not retained on the inactive matrix. And that differential binding you could visualize on gel and analyze by mass spec. You could also run the experiment in competition mode, where you could add compounds to the lysate prior to their challenge on a matrix, and thereby see a depletion of some of these proteins on the active matrix. And by doing this, you'd get some idea of specificity of the interaction, but also by using different classes of compounds, you could see whether the different classes of compounds interacted with the same proteins or not. So on the left here is a gel taken from a simple pull-down experiment. And you can clearly see that there are preferential proteins that are bound to the active matrix that are not present in the inactive matrix. And whilst there appear to be lots of bands, when you sequence these all belong to a single class of proteins, the Bet family of proteins, namely BRDs 2, 3, and 4. On the right here is an experiment in competition mode, and when you add active compounds, not only from a BZD series, but active from other series I'm going to call X here, you see effective competition for these same proteins, suggesting whilst we had diversity in the chemotypes that we'd identified as ApoA1 activators, we actually had a common set of protein targets. So what are these Bet family of proteins, BRDs 2, 3, and 4? Well, they have a conserved domain architecture. They consist of a tandem bromodomain motif at the N-terminus, followed by this ET or extraterminal domain at the C-terminus. They bind to acetylated lysine on histones, and they're part of large protein-protein complexes that result in transcriptional regulation. So these proteins have no enzyme activity. Their sole biological effect is mediated by the fact that they can participate in these multiple protein-protein interactions. Knowing this domain architecture, we then went on to do some transient transfections in HEC293 cells, focusing on BRD2 to start with, and we found that full-length protein, as expected, could be pulled down by a BZD matrix. So too could an N-terminal construct, but not so a C-terminal construct, suggesting that whatever these compounds did, they were localized and interacted at the N-terminal portion of this tandem bromodomain. One problem we had was, whilst this all seemed very enticing and nice, was that there was no known biological activity linked with the BETs with the ApoA1. So there was a lot of skepticism as to whether these observations really could be due to, first of all, specific binding, and by that I mean stoichiometric in nature. Secondly, that even if we got binding, whether they were functionally relevant, were these the targets that induced the ApoA1 regulation, or were they just some random off-target effects? Thirdly, people didn't have compounds that modulate BET function, so how did this work? Did the compounds tighten up a complex that was needed to be transient in nature for the interaction to be functional? Or did these compounds actually disrupt one or more, disrupt one or more of the interactions that the BET proteins participated in? Please shout if you can't hear me. So what we tried to do was try to simplify the system to find out what was going on, to get some answers to these questions. And we started off by making single and tandem bromodomain constructs of BRD2 in a purified state. And armed with these proteins and a tool chest of active and inactive compounds from a number of series, we then embarked upon a biophysical journey to prove specificity of binding. And because these reagents hadn't been generated before, we tested that the N, C, and tandem domain constructs were structured and stable using circular dichroism, which is particularly suitable for these helical proteins. We then used the same technique, but then this time ramping the temperature to generate this thermal denaturation curve of BRD2. When you add active compounds into this protein and then carry out the same experiment, you see that the protein is stabilized, suggesting that there is an interaction between these compounds and the protein. If you add the inactive compounds, you don't see the stabilization, suggesting that whatever we were seeing, this differential interaction that we were seeing in an endogenous system could be reproduced by our purified reagents, giving us confidence to continue. These proteins were then immobilized onto a biocore chip. So what I show on the left here is the immobilization of the N-terminal domain of BRD2, and on the right the C-terminal domain of BRD2. But we also did this with tandem domain constructs. And you see here a series of titrations of the BZD compound over the surface to generate these binding sensorgrams. If you analyze these sensorgrams, what you find is that you have compounds with affinity in the 10s to 200 nanomolar range, and that this binding is specific, as you can see by these kind of tightening of the curves, which shows that the binding is saturatable. The gold standard of direct binding methods is isothermal titration calorimetry, and this technique enabled us to conclusively prove that the compounds bound to the N-terminal bromodomain with 1-to-1 stoichiometry with an affinity about 46 nanomolar, to the C-terminal domain with 1-to-1 stoichiometry of about 52 nanomolar, and to the tandem bromodomains, to both the N and C-terminal bromodomains, the compounds could bind simultaneously with an affinity about 30 nanomolar. And whilst we started with BRD2, we could reproduce these results with BRD3 and 4. So at this point, we were pretty convinced what we had were compounds that were ApoA1 activators, but also ligands for these BET proteins. They bound to the proteins with a stoichiometry of 1 compound per bromodomain, and they had affinities in the 20 tens of nanomolar range. So the next question we set to address is this the interaction that's responsible for ApoA1? And the first clues to suggest that this might be is that on the y-axis here is the increase in mRNA of ApoA1 when you add 1 micromolar of the BZD compound, and you can see that clearly you can induce ApoA1 mRNA expression. You can reproduce that phenotype though if you knock down BRD4. So the first time showing that a linkage between these BRD proteins and ApoA1. Moreover, now we had a clue as the molecular target, what we could do was set up an affinity assay, an FP displacement assay, and what I show on the y-axis here is the affinity of a number of compounds against the BRD4 proteins, but these are actually quite interchangeable, and it would look the same if you interchange that with BRD2 or 3. And on the y-axis here is the ApoA1 activity in a cellular assay. And this shows not only the BZD compounds, of which there are about 150 points here, correlating very well between the cellular SAR and the binding to the BET proteins, but multiple chemotypes shown in grey, so there's about 15,000 spots here, that this correlation is reproduced not only with BRDs 2, 3 and 4, but across multiple chemotypes, really showing that inhibition of these BRD proteins could induce ApoA1. So the third question we had to address was, so how did this work? On the right here is now a kind of familiar structure of a bromodomain with this 4 helical bundle motif. What I'm showing at the top here is 2 loops. In red, the long ZA loop, and the smaller in orange, the BC loop. In yellow is the binding of a histone acetylated lysine peptide, where the acetal of the lysine inserts into a conserved pocket. The rest of the peptide is flanked by these loops. These loops are actually the most variable regions amongst bromodomains. So what you have is the acetylated lysine binding in a conserved region, whereas the loops allow this acetylated lysine to recognize this different kind of peptides. So what about our compounds? Well, X-ray crystallography shows that these compounds bind in this acetylated lysine recognition site. So the mode of action of these compounds is really very clear. The compounds bind in the acetylated lysine recognition site. Their mode of action is clearly to block acetylated histone recognition. They are protein-protein antagonists. And that can be confirmed in a FRET binding assay where these compounds show the ability to displace tetraacetate H4 histone peptide from the active site of these proteins. If we examine the molecular interactions in the acetylated lysine pocket more closely, in yellow here is showing the acetylated lysine peptide itself, and if we just look at the recognition of the acetyl portion, we can see that the carbonyl of the acetyl is recognized by an asparagine which is conserved in 48 out of the 58 bromodomains. The other interaction the carbonyl makes is a water-mediated interaction with the conserved tyrosine at the back of the pocket. And the methyl group of the acetyl sits in a small hydrophobic pocket here. If we look at these benzodiazepine compounds, they're not clearly acetylated lysine mimetics, but they recognize this conserved motif by using one of the nitrogens to make the direct hydrogen bond with the asparagine and an adjacent nitrogen to make this water-bridged interaction with the tyrosine, with the methyl group of the triazole sitting into the small hydrophobic pocket. So you can see now how this triazole moiety mimics the key interactions in the acetylated lysine pocket. But I've already said that 48 out of the 58 bromodomains have this conservation. So how is specificity achieved? If we look at the binding of a histone peptide, you can see that it follows a particular path on the surface of the protein flanked by this ZA and BC loop. We can see that our compounds occupy a different subset of the pockets, and in fact different compounds we found can exploit different subsets of these pockets and grooves along the top. And I'm just going to highlight one area where you can get affinity and specificity, which is this hydrophobic shelf here that we've termed the WPF shelf. Entry into that shelf is given by the size of a residue that we've termed the gatekeeper residue, in analogy to the gatekeeper residue that allows access to the back pocket of kinases. In BRDs 2, 3 and 4, that gatekeeper residue is a leucine or a valine. In other bromodomains, it can be something like a tyrosine. And you can see why that would prevent these compounds binding in some bromodomains. So what we have here is a class of proteins where you get conservation within the key recognition site of the acetyl, but outside of which you can have opportunities for selectivity and affinity. And this is what is ideal for a target class approach. And this is why we believe by finding these compounds, what we've really done is allowed people to explore not just the BET family of proteins, but the entire bromodomain subfamily. So this is all very attractive for structure-based drug design, and what this illustrates is maybe the epigenetic reader domains may be as equally tractable to small molecule intervention as epigenetic enzymes. But we shouldn't forget some of the caveats that we had initially, which is that there are limitations to symbol recombinant screening platform. And these targets, there are only 110 odd residues, and often the proteins are maybe 1,000, 2,000 residues long. They are designed to work in complexes, and within these complexes, the affinity of compounds can differ, and that can be modulated by also the activation state in tissues and the precise complex work that they're part of. So there's a risk here that if you just simply apply a simple reductionist approach, you may get in vitro ligands, but when you try to translate them into a more physiological context, you end up with poor biological efficacy and selectivity. And one way to mitigate that is to combine the power that we use for chemoproteomics with structure-based drug design. And we've done that in terms of an alliance, which is a complementary screening platform with CellZone, where essentially within this panel here, I've shown the industrialization of the competition experiment you saw before using chemoproteomics. And what you do is, rather than assay isolated proteins, you actually assay complexes that have been pulled out of tissue, which means that that lysate that I sort of showed being put down the column could be derived from native cells. They could be derived from cells that are particularly relevant to the indication that you're interested in, cancerous cells, stimulated cells. And this may be a better judge of selectivity. So when we do these experiments with the BZD compounds and other BET inhibitors, we can actually test whether a large panel of these proteins in the phylogenetic tree are pulled back. And what we find is that these compounds do seem to be very selective, even in the context of the endogenous environment. So I'm now going to do the last bit of my talk, which is to do with how these compounds may have an impact in inflammatory response. Now, when you stimulate cells either with TNF or LPS, what you find is that the profile of cytokine induction differs for different cytokines. So you have some cytokines that are immediately upregulated and die down very quickly, so they are due to primary response genes. And you have some cytokines that follow soon afterwards, but there is a lag and they die away much more slowly, and then tertiary response genes that go up over longer periods. And the reason that it's believed that you get these different temporal responses is that these genes responsible for the primary, secondary, tertiary responses have different states of pre-transcriptional regulation. So primary response genes, a colleague of mine has said, is a bit like a car with the engine running, and all you need to do is take off the handbrake and off it goes. Secondary response genes are like a car where you have to start the engine. You have to maybe recruit some factors, for example, these BET proteins. You may have to carry out some phosphorylation before they're ready to go. So if you induce an inflammatory response for LPS and do a genome-wide analysis, you see a large number of genes, about 658, that are upregulated, and this includes cytokines such as TNF-alpha, IL-6, and interferon beta. If you pre-treat with our BET inhibitors and then look at the genes that are modulated, you see that a subset of the genes in green and yellow are upregulated and a subset are downregulated. And what you find is that whilst TNF-alpha is not affected by our BET compounds, things like IL-6 and interferon beta are. So what you have is some kind of specific modulation of pro-inflammatory cytokines. If you examine what these cytokines are doing, what you have is LPS drives recruitment of BRD4 to selective promoters. So our compounds prevent this recruitment, and this is why they block the transcription of a subset of cytokines. If you look at this more broadly, you can see that our compounds actually inhibit the primary response. They don't have any effect on the primary response genes, whereas they inhibit recruitment to the secondary response genes. And that can be rationalized by the fact that in the primary response genes, they may have bromodomains present, and adding the compounds just doesn't liberate them from the transcriptional start sites. They are ineffective. And the secondary response genes, when you add the compounds, what happens is that then you do get a liberation. So they sort of leave the transcriptional start states and you see a depletion. And this comes back to one of the caveats I said at the beginning. One of the worries of looking at these epigenetic reader proteins is that we fear that it might be context dependent, that the context actually would limit our ability to inhibit. And this is what's happening, but in a good way in some ways, because what this has done is limited the compounds not only to a target, but to an activation state, which could be incredibly powerful. So do these compounds elicit a biological activity? Well, I'm going to show you what I think are some dramatic results in an acute inflammatory model, where you give a huge bolus of LPS to mice, and that induces a catastrophic inflammatory event, and they die very quickly afterwards. A number of compounds, including our BET compounds, if you introduce it prophylactically, so in a preventative mode prior to LPS injection, you see that they're protected. That's not very useful in a therapeutic context, unfortunately. More impressive is that if you wait until the mice get shaky and they develop symptoms of shock, and then you administer the compounds, you see that they survive. So what I hope to show here is that these compounds can have real, profound biological activity. So in summary, I think I've gone through that quite quickly, is that what we've used is chemoproteomics studies to identify the BET family as the molecular targets of compounds that are an output of a phenotypic APOA1 assay. That these compounds can target bromodomains that utilize protein-protein interactions, even though these protein-protein interactions are polyvalent, where the natural substrate has only modest affinity, and people didn't think they were druggable. That we have cell-permeable, potent, orally bioavailable small molecule inhibitors of these BET proteins, and that these compounds not only show selectivity amongst the bromodomain family, but also with other targets, and as you see, in a context, probably a dependent environment. And that bromodomains in general are a druggable target amenable to structure-based drug design. I think also some of the emerging technologies, such as ChIP-seq and ChIP-chip and things, are going to be very powerful in enhancing our understanding of the mechanisms of epigenetic gene regulation, because as you can clearly see, it's very complicated and will enable us to better translate from being able to target a protein in vitro to translating it to a therapeutic context. So I think with that, I have a lot of acknowledgements for people that worked on the initial APOA1 program, which is based in Lazily, people that did the chemoproteomics, and this work is being continued as part of Epinova, which is a centre of disease expertise in epigenetics, combined with interactions with people like Sasha Tarkovsky at the Rockefeller. So it only remains for me to thank you.